I would like to to talk about a series of beautiful discoveries which, as already mentioned before, have been awarded this year Nobel Prize in Physiology and Medicine. So these discoveries point towards the existence of a GPS system, so much like the one you have in your car, but inside the mammalian brain. So a system that also we as humans probably use for navigating, so for finding our way in the environment. So this GPS system, so components of this system have been localized in a brain network, which today for simplicity I will refer to as the hippocampus. So I will try to skip the details and try to convey a couple of basic messages. So if you have questions anytime, if I talk too fast, I'm not clear enough, you just stop me and ask a question, okay? Very good. So okay, that's what I want to do today with you. Two things. First thing, we try to find out which signals, so which ingredients we need for building a GPS. So a GPS, like some system which keeps track of position relative to a starting point, okay? And then, second, we try to do an experiment together, and we try to see if we can find these signals inside the mammalian brain, okay? So now we start with the first question. Which signals do we need for building a GPS? So to address this question, I would like to use an example, which I'm sure it's familiar to most of you. So now we try to help Hansel and Gretel not to get lost in the forest, okay? So we try to help these kids to find their way home. So what did these kids do? They were throwing, if I remember correctly, breadcrumbs on the ground, right? Which is a very good idea, but unfortunately it didn't work because of the birds, right? Which ate the bread. So now I suggest the following. So to help these kids, we give Gretel a compass, okay? So Gretel checks in which direction she's going in the forest, okay? And then we tell Hansel, hey Hansel, so from the moment you leave home, you count your steps, okay? So in this example, just an example, 1,000 steps, 1 kilometer. So Gretel checks the compass, Hansel is counting the steps. Now, these kids leave home, so the origin of this axis, like 0, and they leave home, and they go 8 kilometers north. So they go north because Gretel is checking. 8 kilometers, Hansel is counting. Then they go a long way in the forest, 10 kilometers west, and then 3 kilometers south, and now they are left alone. So now the question is, do you think that based on this information, like direction and distance, can Hansel and Gretel compute, so estimate their position relative to home? So do you think that this is a solvable problem? Is it easy? Is it difficult? So I think it's pretty easy. Like you can see from the graph the coordinates of this point, 10 kilometers west, 5 north. So I can also say, based on this information, I think Hansel and Gretel can really get a pretty good idea on where their house is, and they can even compute, more or less, the shortest way leading to home. So this is just an example to show you that you need two basic signals to keep track of your position in the environment. You need to know where you are going, so you need a compass-like signal, direction, and then you need a metric signal, so you need to know how far you are going. So if you have these two basic signals, direction and distance, you can pretty well estimate where you are in space relative to a starting point. Good. So now we found these two signals, compass and metric. We do an experiment together and we try to see if we can find these signals inside the mammalian brain. So we are going to look in the rat brain. So all the discoveries I will talk about today are coming from the rat. So these are very clever, very smart animals. They have a very good navigation system. They orient themselves in the environment very well, much better than me, that's for sure. So we are going to look inside the rat brain. That's the rat brain, more specifically in the hippocampus, the structure that Professor Thier has already talked about. this hippocampus as in its broad definition. That's where it's located inside the rat brain. So, what do we expect to find in the hippocampus? We have neurons, right? That's what the brain is made of. And every time we deal with the brain, we are dealing with electricity, with electrical signals, right? Because that's how neurons talk to each other. So as Professor Thier already said before, neurons emit small pulses, small impulses of current, which we call spikes. So when a neuron is active, it fires a spike. That's the terminology, as shown here. So many neurons active at the same time. So now we need some means for monitoring, so for recording these spikes. And we use a standard technology which the Nobel Prize laureate this year have also heavily contributed for the development and further refinement. So we use very small microelectrodes, and we place these microelectrodes in very close proximity of a single neuron, so that we can monitor with high precision the spiking activity of a single cell in the brain. So we can display the spikes on a computer screen. Okay? So with current technology, you can do these things while animals are freely and happily behaving in the environment. So like they display natural behavior by using flexible cables connected to the recording system. And this is what we are going to do now. So to record the spiking activity of a single neuron in the brain, we are going to actually hear it. So we couple it to an audio monitor. So we are going to now listen to the activity of a single cell recorded in the hippocampal circuit. Okay, so this is what I'm going to show you now. So when the cell is active, you will hear like trrr. When the cell is not active, you hear nothing. Now you're going to tell me which type of signal we are dealing with now. So you see the rat is running around here. The cell is not active. Now it's active. The rat is facing this position. As he looks away, the cell is not active anymore. And then it's active again when the rat faces more or less the same position that he was facing before. So what we saw now, the cell became active when the rat was facing a certain direction. Then it stopped firing when the rat looked away. And then it was active again when the rat kept facing the same position. Okay? So which type of signal we are dealing with here? Any guess? Direction, very good. Yeah, it's much like a compass, right? So the compass always points north. This cell tells the rat where southwest is, right? So it's a directional signal. So we have found evidence that in the hippocampus there are neurons which encode the direction the animal is facing or the animal is running, right? So these cells, which have been called head direction cells, which convey a compass-like type of signal, have been discovered by Taube in the 90s. Okay, so we have found the first signal, compass-like signal. Now, second experiment, we are going to do the following. So every time we are going to record from another neuron in the hippocampal circuit, and every time that the cell fires a spike, we are going to place a dot at the location where the spike occurred, okay? Like shown in this diagram. You see the rat is running around. 3 spikes here, 2 here, 4 here, okay? And now I show you this movie. This is from the Moser laboratory. You will see the rat happily running around. The speed is faster than normal, okay? The rat is happily running around, so we and other colleagues in the field take extreme care that the rats are really happy. That's the only way these experiments work. And to keep them happy, you see the experimenter gives them chocolate. So they like it a lot. And that's what keeps them busy here, to collect chocolate while they run. Okay, you see the rat is running now. The movie is played much faster. You see that spikes here, these white dots happen a bit here, a bit there. There is no obvious pattern now visible, but as the recording goes on, you see that this is not really totally random, is it? So like the spikes sort of cluster at specific location. Now it's getting even more clear. Yeah. So maybe we can stop here. So you see there are some interesting features in this neuron. So for example, the spikes are clustered at the specific locations. Maybe we see this better from the next slide, which is the same cell type. You see spikes happen at distinct locations, so multiple locations, okay? this firing pattern is periodic, so it sort of repeats itself. And do you see any obvious symmetry in this firing pattern? Anything that you see by eyes? Well, you can fit here almost a perfect hexagon. So this is really the most, this is not an exaggeration, this is the most regular firing pattern ever recorded from the brain. You see? These cells have been named grid cells, discovered by the Moser, Maybrit, and Theodor Moser in 2005. So, do you think that the brain could use this system for sort of measuring space? Well, if we superimpose this grid with hexagonal symmetry on the Hansel and Gretel problem, well, now what we will do, we could simply measure the number of times we cross these firing fields. So the firing fields are the location here where spiking occurs. We call them firing fields, okay? So what the brain could do is just count the number of times you cross these firing fields in this direction, and this direction. So the brain could use this metric signal for calculating, estimating the positional coordinates of this point here in space, much like if you are counting steps, right? So this is just to show you very briefly that there are in the hippocampal circuit signals, compass-like and metric-like, which the brain could use for computing an updated representation of our position in the environment, okay? Much like what a GPS system is doing, okay? Very good. Any questions at this point? Everything clear? Good, because I would like to add a little piece to this evidence. So, these upstream signals, which I mentioned today, like direction, grid, border cells, I haven't talked about border cells. It's also a fascinating cell type discovered by the Mosers. Basically, these cells tell the rest of the brain where boundaries or walls are. So these cells get active when we, the rat, and possibly also we, are close to the wall, okay? They tell us where the boundaries are. So what we believe nowadays is that these upstream signals are integrated, so by mechanism that we have very little clue about. So they are integrated and transformed into a place code of space, place cells. This is what Professor Thier has already talked about. Incredible cell type here. You see the cells are active only when the rat visits a certain location, a specific location in the environment. Cells discovered by John O'Keefe. Okay? Now I would like to briefly outline how these signals, and in particular the place cell representation, relates to memory. And to do this, I would like to introduce you to a fascinating phenomenon, but also still very mysterious, which scientists refer to as global remapping. It's a difficult expression. I tell you this with one specific example, hopefully a simple one. Now we do the following. We have one rat which will explore a gray street, okay? So this gray street, and we monitor the activity of 4 place cells, cell 1, 2, 3, 4, okay? So these place cells, as such, they will be active in specific location of this environment. Like, for example, cell 4 might be active here, then maybe followed by cell 2, then here cell 1 is active, and then cell 3. So this is not just an example, that's actually how the real data look like, which I took from this study here. You see in yellow and red where the cells in the brain are active, and you see you have a specific activation sequence of place cells. Like first you have cell 4, then cell 2, then cell 1, and cell 3. Okay? Now, if we do the following, if we take this rat and we put him now on a blue street, yeah? So a different environment, and we look always at the same cells, now the brain somehow realizes that the environment is different and something really fascinating and also deeply mysterious is happening. The place cells now are active in a completely different way, unpredictable. It looks really like random. For example, now cell 3 might be active, followed by cell 1. Maybe cell 2 doesn't even bother to fire. And then we have cell 4. So we have another sequence which is different from the one before, 2104. Yeah? So you see you have two unique sequences, so two unique identifiers which the hippocampus might use for differentiating these two environments and actually for remembering them, because every time that the rat is in the gray street, place cells will fire this way, 4, 2, 1, 3, and when the rat is in the blue street, they always fire this way, you see? So this might be the reflection of a general mechanism by which individual episodic events in our life, not only streets, as shown in these examples, might be encoded in the hippocampus. So this brings me easily to the significance of this work, which, as already mentioned, has been awarded the Nobel Prize for Physiology and Medicine, to John O'Keefe for the discovery of place cells, Mozart's, Maybrit, and Edward for the discovery of grid cells. So these are really groundbreaking discoveries which have taught us a lot about, not only about how space is represented and encoded in the brain, but also about basic mechanisms of memory formation. Very good. So, now, I want to ask the following question. So, I told you now that we as a community, we have learned a lot here, right? And the Nobel Prize is just a demonstration that we have really achieved something outstanding here. Now, the question I want to ask is, so do you think that the story is complete? So, do you think that here we are done? So we as neuroscientists, are we done here and we should do something else? Or are there still things to do? So to put the question in more general terms, so what is the goal of our research here? So what do we want to achieve? I think these are important questions. And to try to address this question, I want to use the following example. So let's assume you are home. You are working on your computer, yeah? And then, poof, the computer is broken. So this happens, right? So something here, I don't know what it is, it's broken. So can you fix the computer at home? So can you repair it? I would expect that you cannot do it. I also cannot do it. I have no clue. And why can't we repair a computer? Because we don't know enough, right? So I think to repair a computer, you first need to know at least the circuit, right? How things are assembled and wired up together. And then with this knowledge, you could then replace the broken components. So I think a similar scenario might well apply to the brain. I think we need to understand the circuits, the actual cells. We need to understand how these neurons are connected to each other in the brain if we want to fix these circuits when they are broken. And this is something that happens, for example, in Alzheimer's disease. You know? So I think if we know the circuit, then in the future we might be able to use this knowledge to repair these circuits and to maybe even restore lost brain function in affected patients. So I think this is our next challenge that we are going to face in the next years, in the next decades, to try to understand the circuit, how is it assembled, the connection between the cells. And we need to make this knowledge available for future medical intervention, so for helping people. And we have already started working in this direction. We have actually developed a method by which we can visualize, we can see the neurons in the brain, which we can see the circuits which form these sophisticated computations I mentioned before, like grid cells or head direction cells. And we do this with a sort of simple trick. So we put a color in our microelectrode. Here it's green, so that when we are close to a single neuron, we can not only record the spikes, but also we make it green, okay? So we can find back the cell we are recording from in the brain. And now I show you just a brief example of our recent work. With this methodology, we could reveal the identity of grid cells in the rodent brain. Most of the cells, not all, but most look like this. This is the structure of these grid cells in the brain. We call them pyramidal neurons. And what we found is that these cells are not present really everywhere, but they are grouped in clusters of cells, which we call patches. You see one such patch contains 200-300 cells, okay? So they are sort of grouped together. And these clusters, these patches are not present only in the rat brain, but also in the human brain. This is ongoing work from the Brecht laboratory, what I'm showing here. You see also in the human brain, you have these clusters of green cells, which look very similar to the ones in the rodent brain. So these are the grid cells. And what about the head direction cells I mentioned before? Well, we find them in another brain structure. Okay, here that's where the green patches are. We find most of the head direction cells here in this structure, which is outlined by the dotted lines here. And it's maybe easier to see here in the cartoon. Here I show you one such cell which we have identified, which codes for direction. Now the cell is colored in red. And what we find, which it's preliminary, but I think it's very interesting, is that these head direction cells send selective connections to these green patches. So we see this at the single cell level, but also at the sort of population level. If now we color these connections with a red tracer, you can see the connections here, which are very specific to the single green patch. So this is just an example to show you, and this is what we believe nowadays, is that there are very selective connections in the circuit which are awaiting to be revealed. This is just the beginning, and I think this is really our next challenge, to try to understand the connection, the circuit, how is it assembled, with the long-term goal of understanding the mechanisms of the hippocampus, and to make this knowledge available for future medical intervention. Okay, having said this is the people I want to thank. So colleagues and friends from the Brecht Lab in Berlin, where I worked before. This is my current team here in Tübingen. And I would like to thank also SIN for giving me the opportunity to continue this exciting research here in this nicely and collaborative and friendly environment, which is SIN. So having said this, I thank you all for your attention. Thank you.